I am Fatinia Kamila, and I have the privilege of being your Master of Ceremony of the occasion. Before starting the event, I will read the rundown for this morning's program, which begins with the opening, then continues with the welcoming speech from Vice Dean of Academic Affairs of Faculty of Agricultural Technology, Universitas Brawijaya. After that, we will go up to the guest lecture session from our distinguished speakers and continue with Q&A session. Next, it will close with a
Good morning, esteemed college student and honored guests. Chào and Nin Hao Sư Chen. On behalf of the Dean Office, it is my distinct pleasure to welcome you to this special guest lecture by Prof. Chaucer from the National Pingtung University of Science and Technology in Taiwan. We are truly honored to have some distinguished scholars with us today. Prof. Chen has made a significant contribution to the biosensors and biomechatronic engineering, and his research has profound impact on biomechatronic engineering. His commitment to the advanced knowledge and fostering innovation aligns perfectly with our mission here at Faculty Agricultural Technology, Universitas Brawijaya. Today, we have the privilege of learning from Professor Chen, wealth of knowledge and experience. His insight on introduction to portable instrument for DNA measurements will undoubtedly inspire us and encourage fruitful discussion among our academic community. We believe that collaboration and knowledge exchange are essential for our growth as scholars and professionals, and we are grateful for the opportunity to engage with esteemed experts like the Professor Chen, and I encourage everyone to actively participate, ask questions, and make the most of this unique opportunity. Thank you, Professor Chen, for taking the time to share your expertise with us today. We are excited to learn from you and look forward to a fruitful discussion. Let us extend a warm welcome to Professor Jyh-Jian. Okay, thank you. Good morning.
Thank you, Dr. Enrica, for your opening. Before we proceed, I would like to extend our gratitude to everyone who has joined us today. Your presence is greatly appreciated. This lecture wouldn't be possible without your support and enthusiasm for knowledge sharing. Today, we are fortunate to have a distinguished speaker with us, Professor Jyh-Jian, or Chaucer Professor from National Pingtung University of Science and Technology, is here to enlighten us with his insights and expertise. I am also graced with the presence of our respected invited guests and our esteemed faculty members who have joined us today. This lecture is a part of the international collaboration program hosted by the Department of Biosystems Engineering, aimed at enriching our understanding of crucial topics within the field of biosystem engineering and broaden our knowledge. Today's lecture will be a stimulating exchange of ideas and knowledge. To ensure the smooth flow of the session, we have a dedicated moderator who will oversee this presentation and the subsequent Q&A session. Next, we will continue with Bapak Pak Darmanto.
Oke, terima kasih. Thank you very much, Mbak Kaira. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Welcome to the International Guest Lecture, which is organized by Biosystems and Departement of Universitas Brawijaya. The Honorable Speaker, Professor Jyh-Jian, Honorable Vice Dean of Student Affairs, alumni, and student partnership, Dr. Muhammad Bagus Hermanto, Honorable Dr. Dimas Firman Aliza, Secretary of Biosystems and Department. I also extend a warm welcome to all faculty members, students, and all participants that join in this guest lecture. It's my pleasure to be a moderator on this exciting event. Our esteemed speaker, Professor Jyh-Jian, our esteemed speaker, Professor Jyh-Jian, is a distinguished faculty member at National Pingtung, Universitas Science and Technology, Taiwan. He received a doctoral degree in mechanical and science from National Chaucer University, Taiwan. And now, Professor Chen is one of leading researchers in the field of heat and mass transfer in microfluidic system, also in computational fluid dynamics in microfluidics, design and integration of microfluidic strips, and portable analysis devices. Prof. Chen is also head of Biosystems Thermofluidic Analysis Laboratory. We are very honored and privileged to have Prof. Jyh-Jian join us today to deliver a guest lecture on introduction to portable instrument for DNA measurement. We will have two sessions here. Prof. Chen will provide a comprehensive overview of the principle and application of portable DNA measurements. It will take around 1.5 hours. We will discuss the latest technological advancement, challenges, and future prospects in this exciting area. And after the presentation, we will have a discussion or Q&A session. Before we go to the presentation of Prof. Chen, as there's a session, photo session for us. Let's we go to the photo session first. Okay. Okay.
Pak Darmanto.
Okay, thank you very much. And now, without any delay, let us welcome Professor Chen to share an inspiring knowledge. Professor Chen, time is yours.
with my students. Because right now there are 3 students in your department, and they work with me during these 3 months. So maybe next time my student can come here to join your team or join your work in your lab. Okay. And right now I'm the head and the professor of Department of Biometronics Engineering. So I am an engineer. So everything I do is that I want to make something more convenient and more useful for everybody. And in my department, we want to integrate the technology, include the biology and the mechanical engineering and the electronic engineering and auto control or the coding inside a device. Okay, today the title of my presentation is Introduction to Portable Instrument for DNA Detection. Okay. Okay. And our topic of our research is build a device for DNA analysis. But the first slide is what is heater connect to DNA? For a heater, we use it when the weather is very cold. But in Taiwan, it's not so cold right now. So last month we have a Mid-Autumn Festival in Taiwan, and many people barbecue near their home, and we use heater. But what is the relationship between DNA and heater? Okay. We go back to how to detect DNA. We got many samples from creation like animal, plants, or people. We got the material from the human body and we want to detect it, the DNA, of course. The first one, we use different methods to purify DNA. And then we want to detect it. But right now, if we want to detect the DNA directly from the human body, we will need a very expensive instrument to detect it. So the very easy way to detect DNA is we amplify it. We use some instruments to increase the amount of DNA. Then we can use a very cheap instrument to detect it. It calls DNA amplification. How to detect, amplify the amount of DNA? We use heater. We use PCR, or we use some isothermal amplification method. Then we can use the traditional gel electrophoresis method to detect it. We can see the band of a gel, then we can distinguish the positive or negative sample. So the most important thing in the DNA detection is amplification, because we can reduce the cost of DNA detection. So for me, I am an engineer, so I want to define or integrate all the functions into one device. Then the heater will be the main component in our device. There are a lot of DNA methods to passivation detection. The application can be used in food safety or forensic detection or clinic test. The pathogen includes like bacteria, fungi, virus, and many methods we can detect the pathogen. But right now, we want to use a correct method and not so expensive method. We want to save time, we want to save money. So DNA may be the main choice we can use. Because for DNA, there is some sequence and ATCG. When we find the complement of the other sequence, then we will make sure the double-strand DNA is correct. Then the detection of a DNA used to this kind of application will be correct. So for the DNA application, there are two main methods. The first one is most popular for many people is PCR, polymerase chain reaction. We use three different and do some thermocycling, and we can amplify or increase the amount of DNA. But right now, not right now, maybe 20 years ago, some research invents some isothermal DNA amplification method. That means that we just need to use one temperature to amplify the DNA. So that may be much easier. But because many people have been used to use PCR, so if we want to change to isothermal amplification, the reagent, the chemical we want the device cannot be reduced to a portable device. So right now we focus on isothermal. Okay. There are several methods, and right now we use lamp and RPA. Lamp and RPA, popular too. Okay. Okay. For polymerase chain reaction, PCR, there are three different steps. We want to increase the temperature and lower the temperature. For the double-strand DNA, and the first step, we will increase the temperature to 95 degrees Celsius, and the double-strand DNA will separate into two single-strand DNA. And we decrease the temperature to 55 degrees Celsius. And then some primer will bind to a complementary DNA. The primer, the purpose of the primer we use is to locate the specified location of the DNA we want to amplify. Not whole the length of the DNA we want to amplify. We just want to specify some location because we will know the DNA sequence will specify for some disease or some animal or some creation. So we just need to amplify this short length of DNA. How to detect the short length of DNA? So we have to specify the location we want to amplify. We use primer. And then we increase the temperature to 72. And with the help of enzyme, some nucleotide will bind to the DNA. And then finally, we will have 2 double-strand DNA. It's called one thermocycling for DNA amplification. And then we use maybe 30 to 40 times the cycle. Then we can increase a lot of DNA. Okay, then we can detect it. It's a basic steps for PCR. Okay, for PCR, it was invented maybe 40 years ago, and after 10 years ago, the Dr. Muller got the Nobel Prize, and the other one is also research for the DNA amplification, and it's Michael Smith, he's Canadian. Okay. And here is he works at UBC in Vancouver, Canada, and this building is to memorize his work. So you can see the structure of the building is like DNA, double-strand DNA. Yes. Yes, I did it there in 2009. Okay, so the application of PCR before is only known by some research or some biologists. But after COVID-19, I think most people know what is PCR because we will detect the virus by using RT-PCR. Okay, so for PCR, we can detect the DNA with the help of PCR. But usually we use the traditional PCR machine to amplify it. It will take at least 2 hours, at least 2 hours. And we want to increase or fasten the time. So we have to choose something or to find some solution. And the main method we use the MEMS technology. MEMS technology is similar to the semiconductor process. So because the semiconductor in Taiwan is famous, so for us to use the semiconductor fab or some laboratory not so difficult. So the MEMS process is also not so difficult for us to do. So why we use MEMS technology? We can build the PCR chips. This chip is not for it. It's a PCR chip. We use it to detect, to amplify and detect DNA. There are 2 main kinds of PCR chips. The first one is we call microchamber PCR. Microchamber PCR is like a small size of a PCR machine. We put the sample inside the device or chamber, and we use the thermocycling, and we can amplify the DNA. So it's like a static reactor. And the other one is we call continuous flow PCR. That means the sample, PCR sample, is flows through different temperature regions. So it can still do some thermocycling, but not in a chamber. The sample flows through different regions. So it's like a dynamic reactor. So for a chamber size PCR, like just a small chamber. For continuous flow, we replicate a channel on the chip. Okay. About maybe 30 years ago, the first PCR chip was invented, and then after 5 years, and the other one is continuous flow PCR chip. So the first one use silicon, because I told you that we use semiconductor process to build the chip. So the first substrate we use is silicon. But the silicon is expensive and not safe for the cell. Okay. So after that we use gas. Gas is cheaper, much cheaper. Okay. Because for the traditional PCR chip, it's just like a small size of PCR machine. So today I will introduce the different one is continuous flow, because the design concept is totally different. For the first one is unidirectional continuous flow chip, and the sample is flows through the channel, and there is several isothermal regions for the sample, and it flows one region and it reaches to the temperature, and it can do some reaction. So after it flows to 2 or 3 or 5 or 4 regions, it can complete one thermal cycle. For design the chip, of course we do PCR in one chip. How we can use different temperature region to do the same thing? 1 or 3 heaters, because we use heat transfer mechanism in the chip. So sometimes we can reduce the number of heater we want to use. Okay. And to design a chip, we have to figure out the most important thing is temperature and the time. For PCR, there is 3 steps I have shown you. The first one is we call denaturation. We will increase the temperature to 95. The second one is annealing step. We will lower the temperature to 55. And the third one, we call it extension. Extension means the nucleotide will bind to the DNA, single-strand DNA. So that means if the length of the DNA we want to amplify is long, we will give then a little time to extend. So for the denaturation and the annealing, we just need a very, very short time. We can do it very fast. But for the extension, we have to slow down. How can we slow down use one single pump? We don't want to change the flow rate of pump. Every time the sample flows through the extension region. We want to use a single one constant flow rate for the pump. It will easy to control it. So how can we extend? We use like a winding rod, okay? Make it longer to travel along the channel. That means we can extend the time for it. So after the extension, we will go to a new one, a new thermal cycle. So we can speed up again, and we can repeat the thermal cycle. Okay, here come to my research. We design different kind of continuous flow PCR chip. The first one we call water cooling continuous flow PCR chip. It's our device, the very old one. We use some syringe pump and some commercial heater, and the next one is the chip we design. Okay. We use a cartridge heater because what we want our size small. So cartridge heater is like a heat roll. And there are 3 different temperatures we want to design. The first one is 95, 55, and 72. And inside it, we can see a tube. Why we use this? We want to use it by water cooling. Because when we use just 3 different heaters to heat or increase the temperature to 3 specified temperature, in the middle part of our chip, it must be, the temperature must be between 95 and 72. It cannot reduce to 55 because of the double conduction. So here, we use the water cooling method to reduce the temperature to 55. Why we use this? Because for the PCR, it will separate double-strand DNA, and then we will let primer to bind the single-strand DNA. If we direct go to the extension, maybe the double-strand DNA separate, and then it will bind again. So the efficiency of amplification will be reduced. So that's why we hope it will, after the denaturation will go to a nearing to let the primer to bind the DNA. Okay, so before we design the microfluidic chip, we cannot just fabricate the chip because we use MEMS process. The process, or the semiconductor process, is very expensive. It's very expensive. We cannot just fabricate it. We have to design the pattern of the chip or pattern of the channel. Okay. Initially, we just use the very simple thermal conduction method. We calculate the temperature distribution of the starting part of our chip. Is the chip size? We can see 40 millimeter to 25 millimeter, not so big. And we can make sure the temperature accuracy in our chip. And we use the material PDMS binding with glass. Okay. And if we want to reduce our size of the device, we don't use commercial heater. We use the MEMS heater. MEMS heater is an electrode binding embedded on the chip. So the thickness of the chip is maybe just 1 millimeter. 1 millimeter. Okay. Or very small. Okay. How can we make sure the temperature is okay? We use a thermal image. We can measure the temperature distribution on our surface of the chip. Okay. And with the help of water cooling, we can adjust the annealing temperature at the mid part of our chip. How or why we want to adjust the annealing temperature? Because for different DNA, we will design different primer sequence for them to amplify the DNA. So for the different primer, the annealing temperature will be different. So for our design, we can adjust the annealing temperature for different DNA and just use the variable of the water flow rate. Okay, we check it with is the traditional gel electrophoresis method, and we use a kit, positive sample kit, to check. Okay, then we go to the second one, TC cooling. In the middle part, you can see it's a channel size, channel width of our channel. It's less than 1 millimeter. It's 95, and 55, and 72. Okay, similar. But right in this chip, we use TC or thermoelectric cooling material to cool down the temperature. Why we use TC? Because we don't want to use another pump to use water cooling the middle part of the chip. We use TC. TC can be used as a heater and also as a cooler. So for TC here, we use the cooling part of the TC. It can reduce the size of our device. And because in this one we also use PDNS and glass bonding chip. For PDNS, the surface of PDNS is hydrophobic. That means the water is not easy or not, the water don't like stick on the hydrophobic surface of PDNS. So we have to make sure we will modify the surface of PDNS. We use 3 different ways to check. FTIR, we check the surface. For the surface, it's easy to bind some protein. And we check the contact angle of the surface. We check the wettability. We use AFN to measure the surface roughness. Okay. The main thing is we want to prevent the protein absorption on the PDN surface, because for the enzyme, for some chemical for PCR, there are proteins. If the protein stick on the surface, then the PCR efficiency will be reduced. So we have to prevent it. Okay. For this one, we check the Q fever virus DNA. And then we want to reduce our chip. How can we reduce it? We use we call it symmetric one heater continuous flow chip. Symmetric means the total isothermal heating region become 5, 1, 3 to 5, and the outer of the chip is for annealing. There are 2 regions, and then extension, and in the middle part is maturation. And we use 1 heater. That's it. We put the heater under the middle part of the chip, and then we use heat pipes to uniformize the temperature at a annealing pump, and also we use it to cooling down the temperature to annealing temperature. Why? Because heat pipe is very cheap, and we just need heat pipe and the fan, and we can cool down the temperature to the annealing temperature. And you can see the size can be reduced to half. And if we, also in the extension part, we also have to extend the time period for the DNA extension. So right now, if we want to reduce the size, we don't want to extend the length of the extension channel. We increase the width of the channel. Then we can also slow down it. And the time period for the extension is not 40 mm. So the total size is reduced. And here we don't use the commercial heater. We use microcontroller. And we increase the liquid part of our channel into our theoretic design. Sorry. Okay. Why we want to increase the liquid part of the chip inside the design? Because we want to investigate the flow rate effects on the DNA temperature. Because the DNA is flows through the channel, so we want to know the temperature is suitable for DNA replication. So we have to simulate the temperature distribution inside the channel. So, because the thermal convection, the temperature of the DNA flows through the channel will be different for the variable flow rate. We find that if we use large flow rate, the temperature will be not what we want. Because sometimes when the temperature, when the flow rate is too large, then the temperature of the sample will not reach the specific temperature, and it flow to another region. It cannot reach the temperature to denaturation, or it cannot reach the temperature nearly, and it flows to another temperature because the flow rate is too high. So the temperature is not suitable for DNA amplification. So for design the RT-PCR chip, not only we care about the channel, we should focus on the flow rate. We must suitable for the DNA amplification. And for IR measurement, we just can measure the surface of the chip. But DNA flows through the channel. We have to measure the temperature of the DNA, not the surface of the chip. So we use the thermocouple insert into the chip. And then we use the thermodynamics to flow through the channel. And the thermodynamics will change the color when the temperature is different. So we can make sure the temperature is suitable for PCR. We also measure the contact angle of the PDMS surface. Okay, it's similar to the Q virus. Okay, and of course we want to reduce the size. We don't need use the heat pipe. We use the TC cooler. The size of a chip is about 40 to 25, 40 to 25. And it's a temperature file, a profile for different flow rates. we can find for higher flow rates of a DNA, the temperature will not suitable for DNA amplification. Okay. And for the DNA amplification, the important thing is if we can amplify the DNA, how can we make sure the concentration of the DNA is enough? We just measure the temperature, we don't know the concentration. But if we want to measure the concentration inside the channel, it's very, very, very difficult. So at least we can simulate the DNA kinetics, and we can simulate the concentration when the DNA flows through the channel. So we use the kinetic equation. Or we use it to help us to calculate the concentration of the DNA, of the double-strand DNA, single-strand DNA, and so on. When it flows through different regions and the velocity we can calculate. And with this equation, we can calculate the concentration distribution along the channel. Okay. Then we can find when the DNA flow into the input, then the DNA is a double-strand DNA concentration. We find the concentration will increase after it flows one double cycle, two double cycle, three double cycle, and so on. And we got a similar result because the flow rate affects the DNA concentration. With a large flow rate, the DNA cannot complete the double cycle PCR, and the efficiency is not very good. Okay. We want to build a portable device, so we have to integrate all the components in one device. So we cannot use the external power. We cannot use external pump. We want everything in our device. So we build our own syringe pump and build it inside the device. Then we can make sure the device can be small enough. Then we use Arduino. We use Arduino Nano to control the driving force of a syringe pump. And then, yes, it's our portable device. Okay. And then we will go to another design, oscillation PCR device. Oscillation is the sample go to this way and come back. Use operation. Okay. So for this one, we built 3 different thermal regions, and we use thermal conduction to conduct the heat from the heater or aluminum block to the sample. Still, we use some simulation method to make sure the temperature is okay. And then we can design it. And we adjust the speed of the sample, moving speed of sample. And we find it's okay. It's okay. We check data. And then we find the result is okay, but the temperature is not very good for us. Because we find if we use the thermal conduction, the gap between aluminum block and the PCR tube, the reaction chamber, there exists the thermal contact conductance. That means there is some thermal resistance between the alluvial block and the tube. Then the temperature must be not exact as we calculate. So we add the effect of the thermal conductance inside our simulation. Then the simulation data will be much fit to our design. Because the thermal resistance it exists for the thermal conduction. No perfect surface for any surface, any material. So we cannot totally erase the effect of the thermal resistance. So we change another thermal source for the heating. We use IR, thermal radiation. We use IR infrared lamp to heat the temperature of our DNA. And then we want to erase or we want to reduce the power consumption. We use the thermal radiation. Why can we use thermal radiation? We check the spectrum of the IR lamp, emission of the IR lamp, and we check the absorption of the water and the PMMA. PMMA is a material acrylic we use to be the material of our chamber. So from the IR lamp, we use the dual spectrum of IR. And for DNA, DNA is soluted inside the water. So most of the solution is water. Okay. So the water, the absorption spectrum is of course near IR. For the PMMA, the absorption is not so strong for the IR lamp. It's a far IR spectrum. So for our design, we want to make sure that DNA can absorb the IR, the thermal energy from the lamp, from the IR lamp. But the acrylic, the material chamber will not absorb too much energy from the lamp. So if we want to increase or reduce the temperature of the solution, the DNA solution, then it's much easier. But the material of chamber will not increase so high or decrease so low. So it will not affect the ramping rate of DNA. So it maybe can increase or fasten the reaction. Okay, it's three different parts or three different locations for DNA. And the time period for PCR we can reduce to 30 seconds. That means if we want to finish all the PCR process, we can finish within 20 minutes. Okay, we check the data. And for virus, most virus is RNA virus. So if we want to use PCR, it amplifies the size of all the molecule of the DNA, not RNA. So if we want to use PCR to amplify the virus DNA, we have to do the RT, reverse transcription for RNA. Then it will change to a complementary DNA, and we can use PCR to amplify it. So for RT-PCR, we have to increase another region for the RT. But That means we need 4 more regions for DNA. So for the automation type of device, we just need specify one heater to do the RT, and after RT process, we increase the temperature to a nearing or to some temperature. Then we can continue the PCR. We don't need to use 2-step RT and then PCR. We can use a 1-step RT-PCR to complete the whole process. Okay, we build our own PCB and check the data. And then right now, I think most students know 3D printer, and many students use 3D printer. Even in our lab, we use 3D printer, and we want to reduce the cost of our device. So we build our own 3D printed PCR device. Okay. Most of them built by 3D printer. Of course, we use laser cutter to cut the ugly plate to make our own device. Why? It's cheap. We can reduce the cost, total cost. And here we use 1 heater, of course. And the heater increase the temperature to 95, and use the thermal conduction mechanism, and we can reduce the temperature to 55 to 72 for the annealing, for the extension. Yes. Of course, for PCR, just for amplification, we have to detect the amount of DNA. For PCR, most of the time we use fluorescence method to detect DNA. Fluorescence method, that means we have to use the heater, use the cooler to do the thermal cycling, and then we want to detect, or we want to amplify the DNA for not only one reaction. We want several samples do the PCR simultaneously. So we make several micro chamber in the acrylic plate. Why? Because we want to do a larger reaction at the same time. We can check the positive or the negative sample. We can compare with the other material, or the other DNA, different DNA. We can do it at the same time. Okay. For the DNA fluorescence detection, after extension or during the extension, there is double-strand DNA exists. For the fluorescence dye, we will bind into the double-strand DNA, and we use maybe blue light laser. Use a blue light, and the blue light laser excites the fluorescence dye, and the dye will emit green light. So we can use some detector like a spectrometer to measure the intensity of the green light. Then we can make sure the DNA intensity is high enough, and we can make sure the sample is good or not. Yes. We can real-time detect during the PCR. Okay. For the bacterial detection, I think if you are a farmer and you own maybe a shrimp farm, AHPND is a very serious problem for the shrimp because it's a fatal disease for shrimp. If your farm got H-E-N-D, caused by viral bacteria. Then almost all things in the farm will be dead. So most or some farmers use antibiotics inside the farm and prevent the bacterial disease. But if this farmer overdose it will cause more severe problem in their farm because of bugs. Okay. So before the disease outbreak, we want to detect it. We want to prevent such disease outbreak. So there are some ways to detect the bacteria. We can use cell culture, we can use DNA test, traditional DNA testing, or ELISA. And it will cost a lot of money and time, and we want to reduce the time consumption, money, and if we can build a portable device like that, it's a point of care testing. It can use in far field, in remote area. It's much easier. Of course, right now, the internet is very convenient all over the world. So if we can use IoT, Internet of Things technology, to transmit the data from the local location to the remote medical center or somewhere, some research don't need to go to the farm to check the data. They can get it without inside the center and easily to detect the result. So use this device. We can purify the DNA. We can use only one temperature. We can use one temperature. Then the amplified DNA will show the result by the color change. If there is no target DNA inside the solution, or fluorescence dye will be quenched, for this dye, it will show the yellow color. If the target DNA exists in the tube, then the fluorescence dye will excitate by the UV light, and it will emit UV light. So we can use our eye to detect or see the result, yellow or green, negative or positive. One temperature means isothermal application. The popular one is lamp. Lamp use one temperature, but it uses 3 pair of primer, 3 pair. For PCR, we use only 1 pair, or two primers. For lamp, we use 3 pairs of primer. Means we use six DNA sequence to locate the DNA sequence. So the accuracy for the lamp is much better than the PCR because it can specify 6 parts of DNA sequence. And it uses only one temperature, 65 degrees Celsius. So of course, it's much easier for me to build a portable device by using them. So, of course, we want to build a portable device. And the first one we use our cell phone to take picture of our sample. And then we use it to analyze the result. Why? Because right now the cell phone is not so expensive. And we build our own app. And if a cell phone, we don't need to buy a very expensive. We just install our app in a very cheap cell phone. Then the total cost will be reduced. In the device, there are 3 main parts of component inside. First one is level control. We use microcontroller, and we use rechargeable battery. We don't use external power supply. And we use two PCR tube as a reagent chamber. So it's much easier for biology used. And we use cell phone to capture of the image of the sample. And we analyze it, use our own app. We design our own PCB, and there are three batteries, rechargeable battery. And we design the graphic user interface. We design the app, and it can do some simple processing for RGB. And there are two reaction chamber inside, so we put the positive and the negative sample to complementation. Okay. So if we don't put fluorescence dye inside, then for negative sample, the color of negative sample is total transparent. If it's a positive sample, then it will become a little white. But sometimes it's not easy to distinguish between two. So it's much easier for us to put some fluorescence dye in it. So we can get the sensitivity of our own device. There is a threshold value if the concentration of the device above 10⁻⁴ nanogram per microliter, then it will over the threshold. So the limit is 10⁻⁴ for our own device. Yes, and then we use 3D printer again. We built our own 3D printed lamp device, and we use microcontroller linked, made in Taiwan, and we also use rechargeable battery, and we use micro spectrometer. We don't use cell phone inside because for me, the size of cell phone is still big. So we use small one, we use spectrometer. Why? Because for the linkage, there is Wi-Fi module inside. So we can transmit information to server, and we can access it by notebook, computer, cell phone, anything, 3D. Something just connect to the server, we can get the data. Okay. So if we got some material and we think it may be infected by something, so we can purify it and put into our product device, and we will do the lamp and also detect the fluorescence dye light, and then it will transmit the data by Wi-Fi, transmit the temperature of RGB value of the image, or the light of intensity to our server, and we can build our own web page, and anything can access it, can read the data. We use microspectrometer, very small, like tip. Size is very small and not big. Okay. So for the lamp device, because it uses isothermal, one temperature, it don't need a very complicated thermal control module. Essentially, it don't need cooler, because for PCR we have a heater and we must have. But for the isothermal, we just need one heater. It will greatly reduce the size of our device. And for the lamp, we can use fluorescence method to detect DNA. We can also use different way. By the colorimetric dye, we just read the color change. It's much easier for the photo device. Of course, if we cannot distinguish between two colors, we can use some microcontroller, microprocessor, with some camera, with some spectrometer to help us. And we don't need to use some very expensive detection. We can use just small camera. We can use Wi-Fi. We can use Bluetooth. We can transmit the data to our device. Of course, right now, not only the lamp is used in the portable device. There is another one called RPA. The difference between RPA and the lamp is the primer for RPA is one pair. That means two single primer to specify the location of the DNA sequence. For lamp, it needs 3 pairs. RPA is similar to PCR, only one pair. So sometimes we use the PCR primer or PCR primer to do RPA, or we use RPA primer to do PCR, much easier. Okay. And the second one is for the RPA amplification. The temperature of amplification is 37 degrees Celsius. For lamp, it's 65 degrees Celsius. For RPA, 37 degrees Celsius. Do you know what 37 degrees Celsius is? How can we reach 37 degrees Celsius? Our body temperature is 37 degrees Celsius. That means if you want to do the RPA without any heater, you just need to hold it for 20 minutes. It will complete the DNA amplification. Finish RPA amplification in your hand. Okay. And right now, there is a very novel one technology, CRISPR-Cas. CRISPR-Cas, before, when it invented, is used to gene editing. About 5 years ago, some research in MIT, America, Boston, America, some research used the CRISPR-Cas technique in DNA detection. It not change total process of DNA detection. It just used it after from the raw sample, and we purify it. And after the PCR or lamp or RPA, after the amplification, we add some enzyme into CRISPR-Cas enzyme into it, and it will help to detect, for us to detect DNA. How can we detect it? Well, there are some methods, fluorescence or color change dye, but the sensitivity will be lower to 10 AM. A is 10 minus 12. Very low concentration of DNA, you can detect it. That means for a portable device, maybe we don't need purified DNA before we amplify it. Because for a pretreatment of DNA purification, it's very complicated. If we want to do a purified DNA process in our device, it will increase the complication of our device. But if we can ignore it, it will help us to design a portable device very easily. So if the concentration of the initial concentration of the DNA is very low, that means we just need a little part of DNA. Not very purified DNA, then we can amplify it in our device. So last year, our group cooperated with a group in Thailand. We use this technology, CRISPR-Cas and RPA. We use microspectrometer to measure DNA, to measure DNA concentration, and we got some results. And for the next few years, we will focus on the detection by CRISPR-Cas. We want to build our own portable device for thermal amplification and CRISPR-Cas inside. Okay. Thank you very much.
Okay, thank you very much, Professor Chen. It's very interesting, very outstanding knowledge for us. I mean, students, I hope you catch what Professor Chen presented, and maybe it's very, very new. However, I think it's very insightful to our knowledge. And we can understand that based on our knowledge using many courses that our learning, I think can get more new design related with the other machine. For example, related with the DNA identification. Okay, now we go to the second session, that is Q&A or discussion session. I have 4 slots here. So, who want to ask or discuss with Prof. Chen, I will give you one. I will give you one. Then, if one student gets more question, it's only one. So one chocolate per one student. Although you have more questions, you still only get one chocolate. Okay. Any question? Any question? Yeah, okay. Please, is there any? Medium. Your name?
Okay, thank you for the opportunity. My name is Bambi. So, it was mentioned before that why we need to detect the one of them is application such as food. Maybe I will ask something related to faculty of agricultural engineering. So the question is, how can PCR be applied in genetic technologies to modify organisms for purpose of improving food production? And what are the potential benefits of using such technology in agriculture? Thank you.
Professor, you can
For example, we have some research before that we want to do it for food safety. We want to detect the adulteration of the food. Because, for example, we use the donkey skin to detect the DNA of donkey skin. Why we do that? Because in China, there is some not food, like medicine, and it's called acetylcholine, and it made material from the donkey skin. And people think in China, they think it well good for their health. But for donkey, it's very expensive. So some merchandise sell it with not really donkey skin. They put some horse skin, pig skin, or some other animal inside. So if we want to make sure if material is from donkey, so we can detect the material for the DNA sequence. We just can make sure if a sequence is matched to the sequence of donkey, not whole sequence. We just make sure the specific region or location of sequence is matched. Then we can make sure the material is belong to donkey. So we can extend it to other food. If some expensive food is, we can buy it in the market, but we cannot make sure the material is true or not, or true or false. Then we can use DNA test to do not, because maybe not very easy to detect, because for the food we buy in the market, we cannot just use it as DNA amplification. If it's a powder, we just can solute it in the solution. But sometimes enough powder, but then we must do treatment for DNA amplification. But if we can use it by some other reagent, and we can solute something in the reagent, it will speed up our process to detect it. Yes. So we can use it in food. Thank you.
Okay, Mas Bambi, you get this one, and this thing. Okay, Professor Chen, you can also ask to the audience for student. What you can give them some question related with your expertise.
Because I'm an engineer and I studied mechanical engineering from college, university, from master's, from 2 PhD. I in Department of Mechanical Engineering. I'm focused on heat transfer. Yes. So for my research, I use a lot of technology from the heat transfer. There are 3 main heat transfer mechanisms I have mentioned. There is only three mechanisms for heat transfer, and we use that in our device. And anyone can tell me 3 different mechanisms of heat transfer I used in our device.
Okay, so do you understand? No. give three kind of heat transfer mechanism that use Professor Chen in the devices. That's correct. Three kind of mechanism of heat transfer. Okay? So your hand. Yeah, I think you have get the course, yeah, the technical course. Very easy, yeah. Only you don't need to explain more, yeah, about the mechanism. You just give the three names of the mechanism. It's transfer mechanism, okay? Does that understand? Want to answer? If there is a student, Mr. Dimas will answer. Okay. Okay, go on.
My name is Ruth Eliayu. The answer is the denaturation, annealing, and extension.
Not in PCR. You can use.
Another process or the steps of PCR. We use some heat transfer material to achieve the temperature in our device.
Yeah.
There is free material for heat transfer.
Yeah. Okay, go on. Okay, your name?
Thank you for the opportunity. My name is Emanuela. In my opinion, the answer is conduction, convection, and radiation. Is that?
Yes.
Yes, great.
Okay, maybe any question from student? Any question? You also get one chapter. Yeah.
Mostly inside device, we use thermal conduction because it's easier to transfer the energy from one location to another location. But for the continuous flow, the sample is liquid, so we need thermal convection. For thermal conduction and convection, we need contact between medium. But for thermal conduction, they have to predict the thermal resistance. So we want to directly hit the sample, then we use thermal radiation. But the application is different because we have to consider the cost, the size of the device. So we use to maintain our device to a stable temperature.
Any question again? Professor Chaucer? To the student.
Okay.
Okay. Before Prof Chen give you some question, if there any student want to ask, okay, please.
Hello, my name is Cynthia. I'm asking for you. Top set for the various type of PCR the sample accuracy. Various from variasi.
The variation of? What is your question? Maybe I can help you.
Top sets for their various types of PCR. Do they affect sample accuracy?
Is the no this give effect of the variation of? I'm sorry, you can
Count.
okay, count.
Yeah.
Kalau Indonesia,
Jalan metrinya.
ya jalan metrinya apa?
Kan ukurannya bervariasi.
Oke.
Nah itu dapat mempengaruhi akurasi kesampelnya atau tidak.
Okay, the cup of the reaction for your person, that is variation of the size. It's correct, yeah. It's an effect to the accuracy of the result.
Usually the sample solution, the volume of the sample solution is 25 microliter. 25 microliter. I mean, for 1 millimeter is 1,000 microliter. 1 millimeter means a cubic size of 1 millimeter, and 1 millimeter. So 1 millimeter, and 1 millimeter, and 1 cc is 1 milliliter. It's 1,000 microliter. So we use 25 microliter. So it's very small. So if we want to keep the temperature of the whole body is uniform, it's not difficult. It's not difficult. So we don't need to care about the temperature of uniform inside of it. So it's easy to maintain the uniformity of the temperature. But if we want to change the temperature, because for PCR we use different temperature, higher, lower. The main difficulty is to speed up the rate of a change in temperature. But we don't just need to change the temperature of the DNA solution. We need to change the whole chamber, of the PCR chamber. So the size is not 25 microliter. It may be if the size of the chamber is 10 cubic millimeter, then the size is bigger. Or maybe bigger enough because we want to do many reactions at a time. So we had to build many reactions chamber inside the chip. So it will increase the size. If the size of the bottom increase, the ramping rate for temperature change will reduce. That's the main problem.
Okay, understand what Prof. Chen said before. So we need, if we want to make a speed up the reaction, we can more chamber in the devices. Okay, that's your chocolate.
So that's why for PCR device, if we want to make it portable, we must pay attention to the ramping rate of the temperature. So for portable PCR device, it's much more difficult because the thermal control margin. But for isothermal amplification, we just need one temperature. We don't need to care about the ramping rate. We just increase the temperature to 65 or 70, 37. Then we will keep it stable at one temperature. So for the portable device, many research focus on the isothermal amplification. They don't use PCR in a portable device. Yes.
questions? We have still 2 chocolates here. So we need 2 students to ask or to discuss, or maybe some students want to discuss with in the design of the devices, maybe how to make, for example, how to print, what is device to make 3D printing. What is the type, for example? You can also ask like that. Okay, any questions again? Okay, if there is no students want to ask or to discuss, maybe a second chance for Professor Chen to give you question. The second question from Professor Chen.
And there are several DNA amplification methods, and the important thing to amplify DNA, not only the DNA sequence, because the sequence is very, very long. We cannot duplicate such long distance of DNA in a lot of time. So we just amplify the specific region. We just need to know this region is, compared with other DNA, is unique. Then we amplify it. How can we identify the region? We use primer. Okay. So for PCR, how many pairs of primer we use? For lamp, how many pairs of primer we use? Okay.
2 questions. For PCR, how many lamp that we use? And how many pairs that we use? Okay. 2 students. Okay. For PCR, how many pairs that we use? And the other is for lamp. How many pairs that we use? 2 students. Okay. 1 student. Although you're wrong, I will give you chocolate.
Okay. I'll introduce myself. My name is Ria. For lamp, the lamp primer can conduct the 3 pairs.
3 pairs, correct?
For lamp, yes.
Yeah, okay. We give applause for this question. The last question. There's still 1 question that's not be answered. Okay, go on. Okay, Mas siapa? Although you wrong, I will give you chocolate.
In the PCR, there's 2 pairs.
Only 2. Only 2, correct? Professor? Still not correct.
You mean 2 pairs or 2?
2 pairs.
2 pairs.
2
2 pairs means 4.
2 pairs is 4. Yeah.
2 pairs means 4. So you mean 2 pairs or 2?
2 primers.
2 primers.
Okay. Correct?
Yeah.
Yeah. Okay. Okay.
For PCR, 1 pairs, 2 primer. For lamp is 3 pairs, 6 primer.
Okay. Okay, Professor Chen, I think the session is enough because students will continue to the class today. And let's give us a round of applause to Professor Chaucer. We Chaucer can come into Brawijaya University again and share with other knowledge that's very insightful. Yeah. Okay. I think it's enough. Today I will give the close session to Mbak Kaila. Okay. Professor Chaucer. Thank you very much.
Thank you, Moderator Pak Demarco. Thank you, Prof. Chaucer, for sharing your expertise and insights with us today. We are truly appreciate for your time and also your knowledge. We extend our heartfelt gratitude to our invited guests and our faculty members for their presence here today. Ladies and gentlemen, that concludes our international guest lecture today. We hope you found this lecture enlightening and inspiring. Once again, thank you very much for their outstanding contribution. Thank you all for joining us. Have a wonderful day.